Welcome everybody. This is a new video about my latest publication for BioEssays. And as you can see, I'm wearing a tie, and that means that I consider this one an important one. And let's hope that you agree. So what's this video going to be about? It's entitled Changing Gears with ACAD9. And I'm going to share the presentation with you to talk about what it's all about. Changing gears with ACAD9. And it's about ROS damage, so reactive oxygen species damage in the mitochondrial respiratory chain upon the change of using a different substrate. So going from glucose to fatty acid oxidation, for instance. And I want to dedicate both this video and my article to the people that really did the experiments. I only interpret their very nice results generated by a lot of effort. So the real title is Molecular Characteristics of the Multifunctional Fatty Acid Oxidation Enzyme ACAD9 illustrate the importance of FADH2 NADH ratios for mitochondrial ROS formation. And now you understand why I preferred the title Changing Gears with ACAD9. That's much more catchy, and this sounds so very complicated. But actually, it's less complicated than you think. And to explain why, I'm going to have to go back in time. And back in time means to about to 2010 and 2011, when I wrote up a hypothesis to explain really strange different aspects of eukaryotes and the functioning of certain tissue types for a very high level eukaryotes such as ourselves. And I thought that you could explain the ubiquitous existence of a tiny organelle in all eukaryotic cells, whether simple unicellular eukaryotes or very complicated multicellular ones, such as animals, and the fact that our neurons don't use beta oxidation, that these could be explained by the same basic aspects. They both have to do with beta oxidation. And what is the link there? The link is that you generate way more ROS, according to my model, when you catabolize fatty acids instead of glucose. And that's nicely illustrated on the cover of the BioEssays issue in which this hypothesis article appeared. And you can clearly see the difference. Over here, it's glucose using just two different complexes, complex 1, NADH dehydrogenase, and complex 2, succinate dehydrogenase. And electrons flow in. But in the case of beta oxidation, a lot of electrons flow in via an extra enzyme, the ETF, and that's linked to the first step of beta oxidation. And now you can see that this leads to a whole lot of covering of the acceptor of complex 1 with electrons, so there's no more free space and you get ROS formation in complex 1. And that was the basic idea. Now, I extended that model with ROS formation in complex 3 and put in a lot of extra nitty-gritty to explain what's going on. And if you're interested in that kind of nitty-gritty, then you should read the 2009 publication that I indicate here. But that's just to fill up the molecular details. The basic idea actually is pretty simple. It's like a traffic jam of electrons. And if you have a traffic jam, then further upstream, you can have collisions. And these collisions are the ROS damages in complex 1. Now, when I wrote up that article in 2011, after it had been accepted, an article appeared by Jessica Naus and Leo Nijtmans. Leo Nijtmans, a friend of mine. And they demonstrated something really unexpected, that a close homologue of the normal very long chain acyl-CoA dehydrogenase ACAD9 actually was a complex 1 chaperoning. So it helped to make complex 1, which is a very complex, you could say. And it needs cofactors to get synthesized. And I mentioned that in the article, but there was one thing that made it a little bit less exciting, I thought, and that was the fact that ACAD9 didn't seem to be a real beta oxidation enzyme anymore. But over the last 10 years, a lot of new research has been going on, and these lead to rather exciting new insights. First of all, it was shown that ACAD9 really is an active beta oxidation, fatty acid oxidation enzyme. And based on its substrate preferences, it could even be a rate limiting one. So ACAD9 could be a factor that determines how much beta oxidation is going on. That ACAD9 really is an essential part of the mitochondrial complex 1 assembly machinery. Is also been shown in multiple ways, so everybody is really convinced about that now. And that these two functions are mutually exclusive is also sure. And why are they mutually exclusive? That's regulated by the loss of the FAD cofactor as soon as ACAD9 loses its FAD cofactor, it changes conformation, can't do reuptake of that fat cofactor anymore, and becomes a real constituent of the mitochondrial complex 1 assembly machinery by linking to exit due to this conformational change. A lot of the results that I talked about here are presented as a way in which people say, yeah, of course, you now need more complex 1 because you don't do beta oxidation anymore. But that I find a rather naive way of describing things. Because in the case of beta oxidation, you also generate an enormous amount of NADH. So probably it's much better to look at this, that you have to repair the damage in complex 1 by, you could say, after sustained beta oxidation. So that makes much more sense. And there's also a more hypothetical aspect. The FAD that is lost by ACAD9 could be physically changed by hydrolysis from a flavin adenine dinucleotide to a flavin mononucleotide. And that's precisely the cofactor that complex 1 uses. And that's shown in this picture that comes from the latest article. So we are absolutely convinced that this is true upon loss of its FAD, shown over here. This changes conformation, can now attach itself to exit, and this forms, together with a third factor, the core of the chaperonin complex that allows the synthesis of complex 1. The second part, so that's shown over here, the second part is still hypothetical, but there are enzymes that could hydrolyze in mitochondria this flavin adenine dinucleotide into the flavin mononucleotide, which is the cofactor of complex 1. So that's really complete repurposing upon damage by this factor that was involved in the process that had the ROS damage originating. So if you find this exciting stuff, then please watch and read the other videos and articles in BioEssays that I published over the years. And please do that, because there is a large need on my side for real criticism and real insights. And to be totally honest, it's also about something else. I'm a really needy scientist, and I haven't reached the magic number of 1,000 views for any of my videos yet. So could you please help me out here? I know I sound a little bit desperate, but that happens when you're really involved in a certain subject. So this is what it's all about. I hope you enjoyed the presentation, and I look forward to your comments and feedback. Bye.